My name's Chris Deich. I'm a graduate student in Kate Adamala's lab at the University of Minnesota, and I'll be presenting on our research towards the construction of a synthetic cell based on Mycoplasma mycoides. So basically, to this day, researchers are very fascinated by how life emerged on Earth from non-living matter. A synthetic cell, we think, is capable of coupling DNA, RNA, and protein synthesis in a contained vesicle, and has the capacity to replicate itself. And a synthetic cell like this has the potential to bridge our understanding of how life evolved from a combination of less complex components. And beyond connecting non-living and living matter, and providing plausible theory for the origin of life. Synthetic cell research also provides a way to investigate internal cellular processes and allow for improvement or addition of new functions. Kind of some of the engineering approaches on the left here, the bottom-up engineering approach seeks to increase your complexity by stacking many systems of non-living components together, attempting to make a replicable synthetic cell. This bottom-up approach has the disadvantage, though, of it doesn't start from a living template, such as the way that a top-down approach would. And so the actual genesis of an artificial cell from these bottom-up components can be quite difficult. However, an example of this, like a bottom-up system, would be the pure system. So it's non-living components. And they basically isolate the transcription translation machinery of E. coli and reconstitute that with the necessary transcription factors. And then this basically gives you a soup that's ready to do in vitro protein synthesis from a DNA or software input. But looking at the top-down method, it seeks to reduce that complexity of a living organism down to only the minimal set of genes required for life. And the top-down approach has this advantage of starting from the genomic template of an already living organism. So you already have a little bit of a roadmap. And an example of this would be the development of the JCVI Syn 3.0 genome from Mycoplasma mycoides. So it's already one of the smallest bacterial genomes, as we've been talking about. It's an obligate parasite. And as previously mentioned last week, they used transposon mutagenesis, and they were able to reduce the genome down to 531 kilobase pairs, 473 genes. And then they were able to basically successfully transplant this back into a cell and have the daughter cells divide and isolate cells that just had their synthetic genome in it. And so we've already demonstrated, or previously, they've already demonstrated JCVI that genome transplantation, but the aim of our research is to take a step further and create a synthetic minimal cell without the need for genome transplantation. So basically, we have the software made for us by JCVI. This would be the Syn 3.0 genome, which is already much smaller than other genomes, so less complex. And a cell-free extract made from that minimal genome could be paired with this synthetic genome to reconstitute a minimal cell. And the creation of Syn 3.0, like I said, Syn 3.0 genome is analogous to your software used to boot up a computer. We need the creation of compatible hardware to boot up the synthetic cell. And so that requires a cell-free extract made from a phylogenetically close relative. And presumably this is because the software of your donor genome has to be capable of interacting with the machinery and the recipient cell cytoplasm. So for this reason, we chose to prepare extracts from NCAP and Syn 3.0 for pairing with the Syn 3.0 genome. And so basically most efforts for construction of synthetic cells are based on E. coli cell-free extract systems, either using lysate preps or pure, like I previously mentioned, that protein synthesis using recombinant elements. And it's E. coli is a very valuable model. We see it all the time, but the organism is vastly complex compared to Mycoplasma. So the extensive genome reduction that we saw with the Syn 1.0 that led to Syn 3.0, this is basically 531 kilobase pairs, 473 genes. It's a lot simpler than E. coli, and for that reason, we thought this would be a good starting place. And our aim was to create this cell-free transcription translation system capable of protein synthesis, and our plan was then to construct a synthetic minimal cell from non-living parts by filling micron-sized lipid vesicles with the cytoplasm derived from the mycoplasma bacteria, along with the genome that we installed, the software. And E. coli, like we said, cell-free extracts are the best. They have the best protein production levels and the most well-established protocols for the lysate preparation. So we decided to go with that, and we used a paper from Sung et al., which basically used the same protocol and was able to generate functional extract from a number of various bacterial species, including gram-negative species such as B. subtilis. So basically, for the preparation of this, cells were grown in SP4 media because they're obligate parasites. They need this gold-plated media. They were harvested at stationary growth phase, which was indicated by a pH indicator at pH 5. They were then centrifuged down 5,000 G for 15 minutes. We isolated our pellets, and then they were washed several times with S30 buffer. That lysis was then achieved using sonication, French press, osmotic lysis, or grinding the cell with liquid nitrogen. And after cell lysis, the cell extract was isolated and underwent a runoff incubation to release ribosomes from their transcripts, and then dialysis was performed in the S30 buffer after that. Then finally, these extracts were flash frozen, and the viability of them was measured using fluorescent reporter proteins or fluorescent RNA aptamers. I don't think I need to say anything about fluorescent proteins, but fluorescent RNA aptamers is just a single-stranded RNA oligo that can fold into secondary structure that, when it binds its ligand and hit with the right wavelength of light, it can give off a fluorescent signal. So a good way to measure transcription. And this is again just looking. This is just a little overview again of lysis methods. We use not only sonication at a number of different amplitudes and cycles. We also used osmotic shock, 250 millimolar NaCl. We tried liquid nitrogen grinding, which was just basically grinding the pellet with continuously pouring on liquid nitrogen for about a 20-minute period. We used a French press, which is just using pressure, and surfactant-based methods as well. And kind of just to give you kind of a nice visual of all these different methods that we use to produce extract. This is kind of a Sankey graph mapping out some of those things, different centrifugation forces, with and without runoff, with and without dialysis, with and without flash freezing. But yeah, we basically threw the kitchen sink at the lysate, trying to make functional cell-free extract. Beyond just the lysis methods, we also, on the right here is the optimal E. coli cell-free conditions with your cell conditions, DNA template, lysis buffer, dilution solvents, things like that. The amount of lysate fraction, 33%. And this is all from the literature and what has been shown to be optimal over time. And this is on the left here, what we tried, the different ranges for our mycoplasma cell-free extracts. We also, this is just again, just a layout of the reporters. So fluorescent aptamer, GFP or M-cherry, basically used to see how functional our lysate was. And now we get to the actual data, and this is, unfortunate, but we were not able to generate any optimizable signal. I have data here. This is specifically MCAP with the osmotic lysis versus some of the sonicated lysates. And this 200 fluorescence is pretty paltry and obviously much, much lower, and not much difference between positive and negative here for the osmotic lysis. Liquid nitrogen grinding, these methods were used on both Syn3A and MCAP as well as E. coli, but E. coli unfortunately was the only one to generate any appreciable signal. So basically showing that the principles of these protocols work for E. coli, but for some reason not for mycoplasma. And again, this is just more data. There's tons and tons of this where we just don't see any signal. This was just different sonication energies, different either using phospholyl pyruvate or 3-PGA as the energy mix, runoff versus no runoff. And really we get no signal. This is just a fluorescent standard, this blue 10 micromolar fluorescein here. So it's not an actual signal from one of our cellular extracts. We used to normalize signal, but overall just showing that we weren't able to get any real optimizable signal from fluorescent proteins. So at this point, we decided to try fluorescent aptamers to see if we could see any sort of level of transcription. This data on the left here is basically just this data on the right without the negatives next to it. And again, we're not really seeing any level of in vitro transcription using the malachite green aptamer. E. coli might come up a little bit here, but really these numbers don't really tell us too much about, the signal to noise there is not very good. And I don't feel comfortable going off, making any conclusions off that. But overall, just a lack of transcription going on in our lysate. And so this made us curious, further curious why we aren't seeing any transcription. And so we wanted to see if I took already previously transcribed aptamer and incubated it with its ligand in a folding buffer, and we added it to our mycolysates, what would happen? And basically that data is shown here. These are different sonication energies here. And as you can see, compared to E. coli, these seem to drop off pretty precipitously over time. This is SYN3A versus MCAP. And again, SYN3A doesn't seem to fall off as bad as MCAP, but it's still falling off quite a bit. And these are just basically kinetic representations of the same data. This one would be sonication, and we don't see as much activity with the degradation here, but when we look at the liquid nitrogen grinding, which is effectively the most gentle lysis method that we can possibly use, you can see SYN3.A is degrading quite quickly. This purple line here, you have E. coli, but even dropping even more precipitously here, we have MCAP. So basically, whatever fluorescent aptamer that we're putting in there is immediately being degraded, and we're not seeing any signal. And then one of the last things I just want to show you guys, one of my collaborators is going to kind of finish up the second half of this talk and where we went with this. But we did look at using a bioanalyzer to look at different transcript, the abundance of different transcripts at different lengths. Basically, we see before lysis, you can see the 23S and the 16S ribosomal subunits and the RNA pertaining to them. But after lysis, that is lost immediately. And after runoff, anything that was even down here, like the 5S, is completely getting washed away. These are basically just really, really small fragments of RNA nucleotides. And so this again is pointing at the second that we are losing compartmentalization of the cell, we're getting a breakdown in RNA occurring. So in conclusion, the SYN3 mineral genome offers a simpler platform for the creation of a synthetic cell. The E. coli model organism is a model organism, but it's vastly more complex. The lysates from MCAP and SYN3.0 didn't produce optimizable TXTL. We saw RNA aptamer signal degraded quite quickly, and then RNA ribosomal subunits were being degraded during immediately after decompartmentalization of the cells during lysate prep. And so from here, we did go further, trying to kind of analyze exactly what the culprit was for this RNA degradation, but I'm going to save that for my collaborator as he goes more in depth on that. I did want to just say thank you to the collaborators and funding. Yeah, it's kind of an awkward place to stop, but I think more of your questions will be answered as we go into the second half of this presentation. But if you guys have any questions, feel free.
Chris, what if we wait on questions and you and Andrei show yourselves after his talk, because these
that'd be good.
the next talk is very much a companion talk to the one you've just presented. So Andrei, why don't you, Chris, stop sharing, and Andrei, you're up.
Yes.
Sure. Okay.
Here, 1 second.
0.
Okay. Are you guys able to see my screen?
Yes.
Yeah.
Go to presentation mode, please. Yes, great.
Yes, cool. Okay, so I'm just going to follow up what Chris was talking about. So I think he just gave a great introduction about our cell-free system, and I will just follow up with the troubleshooting, in fact, because what Chris just told us about is that we cannot see any transcription, even transcription we cannot see. So I will show you guys a little bit of an overview of how we are troubleshooting the system. And just before that, I want to give a general explanation about the projects that we are developing here in the Netherlands in the Red Bull University. So our lab is trying to build also a synthetic cell as Chris and Kate in Minnesota. And the way that we are doing this is, of course, a combination of top-down and also bottom-up approaches. So we basically starting from living cells, and we isolate genomic DNA and isolate also the lysate. And then we put those parts back together in the cell-free expression reaction system. And once we have this whole system with these components optimized, we encapsulate those into liposomes and try to manage the genome rebuilding, like how can we control the expression of the genes inside the genome. And hopefully in the end, we can rebuild a synthetic cell. And what we've done so far, we are working also with the E. coli as a model organism. And we basically already optimized this the cell-free expression system and also the liposome encapsulation of the E. coli genome. And we can see expression from chromosomal genes from this genomic DNA. The idea in the end is that we can use the mycoplasma as a model organism because of many advantages. The mycoplasma genome is much smaller than the E. coli, so that solves a lot of engineering problems when it comes to liposome encapsulation, for example. And the minimal metabolism also we see is a very attractive feature for the mycoplasma. Have been said that, so this is just some results that we got for the genomic DNA from E. coli. So what we show here, we isolate the chromosome from the E. coli, and it already has a GFP gene in the chromosome controlled by T7 promoter. And then we just encapsulate it into liposomes. We have a microfluidic system established here in the lab, so we can use that for encapsulation. And you can see here that over time, we get a signal of GFP expression. So here in this graph, we show the GFP signal increasing as also the size of the liposomes decrease, because here we use some sucrose solution to shrink the liposomes. That gives more stability to the liposomes. So, okay, so I'll come back to our results for the mycoplasma system. So what you've seen so far is that we don't get any transcription from our lysates prepared basically by two strains. We worked with the mycoplasma capricolum and also the SYNC3A. And the first thing that we wanted to know is like what is the poisonous component in the mycoplasma lysate? What is going wrong with the mycoplasma lysate that we don't get any expression? So we designed this assay using the E. coli cell-free expression system because we know that it works well as a positive control. And what we did here is we replaced basically the E. coli lysate in this cell-free expression system with the mycoplasma lysate. So as you can see here in this middle graph, we just have increasing amounts of mycoplasma lysate in the E. coli cell-free expression system. And as we increase the fraction of mycoplasma lysate, you can see that the expression of GFP depletes also decreasing over time. So for sure, we saw with this experiment that there's something wrong with the mycoplasma lysates that's just shutting down transcription and translation here. And we know that by literature data that mycoplasmas are parasites or obligate parasites, and they need to scavenge nutrients from the external environment. This includes also nucleic acid. So one of the hypotheses that we had in the beginning is that probably nucleases are involved in this as a poisonous component in this mycoplasma lysate. And what we tested then is that we found that calcium chloride would be able to deplete a little bit of this nuclease activity in Mycoplasma capricolum. And this is the reference for that. So what we did then is we added calcium chloride into this system in which we have half Mycoplasma lysate and half E. coli. And what we saw here is that we got a recovery of the GFP expression. So this indicates us that probably the culprit of this disabled transcription and translation could be nucleases. So what we wanted to know later is also, is there any way that we can remove these surface nucleases from our mycoplasma cells? So here, what we try to do is just treat with trypsin our cells or Mycoplasma capricolum or SIM3A in order to reduce, at least try to reduce the amount of surface nucleases, and then have a more functional lysate in the end. So what we did here, we prepared these trypsinized cells, and from these cells, we prepared the lysate. And we tested these lysates in a similar way that I showed before, mixing with E. coli lysate. And what we see here in this middle graph is that there is no inhibition of the expression until more or less 75% of this trypsinized lysate. And the expression profile that we see here for this trypsinized lysate is very similar to what we observed to our negative control, in which we just dilute the E. coli lysate with the buffer, in this case, the S30B buffer. So what we saw here is that this trypsinization can help reducing the nuclease activity in the lysate. And in this case, for this trypsinized lysate, we saw a small amount of transcription. So here is the expression of this spinach 2 aptamer over time. So we can see transcription, but we were not able to see translation yet. So we kind of started fixing our system, but not completely. And then later, what we wanted to know is that what is happening to the content of our RNA inside the lysate. Because, for example, if you have a degradation of ribosomal RNAs in the ribosomes, of course, it will affect your translation. So what we analyzed then was how was the RNA content of our lysates throughout the lysate preparation method. So here in the bottom is just the RNA content of the whole cells before the lysis. Here is right after the lysis in red, and in blue after the runoff incubation, and in green here is after the dialysis. So what we can see here is that right after the lysis, either by sonication or by French press, we were not able to see any of those long ribosomal RNAs, the 16 and the 23S, were just absent here right after the lysis. So that also explains a lot why we don't see any translation in our CFE systems. We observed that this behavior also for the trypsinized Mycoplasma capricolum lysates. So it means that the trypsinization also helps to reduce this nuclease activity, but not removing it completely. Next to what we wanted to see is what is the source of this RNase activity? Does it really come from the surface of the cells? And then we developed this assay in which we tested the capacity of Mycoplasma capricolum and SIM3A cells to degrade exogenous RNA. So For that, we just isolated the cells from Mycoplasma capricolum and Syn3A by a sucrose cushion, and we mixed E. coli lysate. And then we analyzed the final fractions of RNA. And what you can see here is that when we add this whole entire cells of Mycoplasma capricolum, you don't see anymore the ribosomal RNAs. So it means that we have a very strong nuclease activity coming from the surface of the cells. And the same for the trypsinized cells. We can reduce the nuclease activity, but not completely, because we can see here degraded ribosomal RNA also for the trypsinized cells. What we see differently, though, is that for the Syn3A, we see a lower RNase activity. So when we add the Syn3A cells to the E. coli lysate, we are still able to see some peaks here for 16S and 23S. So it's just that for Syn3A, the RNase activity is lower. And after all these trial and errors, we just try to test very different methods, because so far we just worked with these physical cell disruption methods with sonication and French press, and we just wanted to use another method, and we just tried surfactant-based methods, which are not very common for cell-free expression systems. And what I'm showing here is we used this digitoning, which specifically precipitates sterols. So it works well for the cholesterol-containing membranes of mycoplasmas. And the thing here is that digitoning will not completely lyse the cell. It will open pores in the cells, and the cytosol can leak out. And what we observed here is that when we use digitoning, we can recover a higher amount of ribosomal RNA in the final lysate, which is a very exciting news. So we run the lysate into this denaturing agarose gel, so we can see that the ribosomal RNAs the profile is very similar to what we see in the E. coli lysate. But again, here we tested transcription and translation, and even for this digitoning lysate, we cannot see translation. So we still didn't solve the problem, but I think we are probably getting our way to solve it. Yeah, so my final conclusions, we find out that even the ribosomal RNA degrades at the cell lysis, so we have a very strong nuclease activity, and we found that digitoning helps to preserve this the RNA content. I would say that our challenges then is how can we optimize these transcription translation reactions using these digitoning lysates, and also for future experiments we're working on how to improve the reproducibility of experiments using genomic DNA. And of course, controlling the expression of these genomic DNAs for building these synthetic cells. I would like just to thank everyone collaborating in this big project, and I will be happy to take questions from you.
So, folks, let's address questions to Andrei and Chris. And we are speaking for our consortium. If anyone has any experience using cell-free systems in mycoplasma, I can't find it in the literature, but maybe someone listening has, and we would love to hear from you if that is the case. I'll ask a question. So the reviewers of our paper that was, they asked us to revise, one of the reviewers was not convinced that RNase was the problem. He didn't say what he thought the problem was, but still can any of you imagine, or Chris or Andre, can you imagine what else it might be if it wasn't RNase?
Yeah.
Oh, I have no experience at all with cell-free and mycoplasma, but with some other organisms. Have you tried different backbones? It looks a weird question, but we noted that some proteins do not, in the cell-free system, we are unable to express, and different backbones made completely different. Some different backbones we were able to express protein that with some other backbones. It doesn't make a sense from the expression, but anyway, the question is, have you tried different backbones?
You mean like using different DNA templates for coding for different proteins? Is that what you mean?
Yes.
Okay. No, so basically we are just using these fluorescent reporters. We are using this M-Cherry reporter and also GFP. We did not try any other ones, but we know for sure that, for example, the M-Cherry works well. The sequence has been previously optimized for mycoplasma by Yo Suzuki in their 2019 ACS synthetic biology paper. So we know for sure that at least for the codome perspective, it should work well. But yeah, we didn't try any other types of genes.
I'm not focusing in the codifying sequence per se, but in the backbone, which actually holds the codifying sequence and promoters and terminators and so on.
Yeah, I don't think we made any concerted effort to try any different backbones. That would be an interesting idea, trying to put our codon optimized M-Cherry and maybe some different backbones, different vectors.
Yeah, I would suggest that. It's not difficult, so it's a simple experiment. I mean, it's worth trying.
I was just wondering, thinking about the question that you asked John about other hypotheses that the reviewer told you. I was just wondering, maybe because we are talking about minimal cells, wouldn't it be interesting as an hypothesis to think about the stability of all those complexes that we are playing with? As you know, for example, ribosomes of mycoplasma, they've got some subunits that are not there anymore. And well, I was just wondering, what vision do you have at the moment of the stability of all the complex that we are used to play with when we play with E. coli cell-free extract?
I would say that because it's such a complex system that we have so many components in there, and we don't really have an output yet to know how healthy is our system. So it's a bit difficult to choose or narrow down which components we should test. So I think that's one of the biggest challenges that we have. What we've done so far is just from the standard protocols from E. coli, we are trying to adapt that whole method to the mycoplasma system. But yeah, I think the sources of like things that we have to optimize there are so many now, and this is something that we are struggling with, I think. Chris, I don't know if you want to add something.
Yeah, I mean, I know we also add exogenous ribosomes as well to kind of bolster the system. And I know those work very well in E. coli. So it's something about adding these components to the mycoplasma lysate that is causing them to theoretically break down. I think for me, the nuclease story works so well just because we're like, after decompartmentalization of the cell, we're seeing all of these things get degraded, and we can, reduce the severity of those things by, say, trypsinizing the outside of the cell, where you might have a surface nuclease, something like that. So I guess it's hard for me to look past that to what else it could possibly be, when it again, like Andrei said, it's hard to actually individually test any of those things.
And do you think that, I completely agree that your data are completely consistent with the RNase story, but I was wondering how could we explain that such a RNase would be mandatory for the cell to survive? Do you think this is the case? And so it would mean that we are talking about, basic function of the cell. And so how can you, how can you adjust this with the fact that it would be a surface RNase? I mean, if it's, if we are talking about a RNase which is involved in the interaction with the host, I can understand this, but I don't see this kind of RNase as, playing with the basic reactions in the cell for translation and transcription. So, so it should be possible to inactivate this one, no?
Exactly. I think there are many nucleases that have been removed during deminimization to SYNC3A. That's for sure. And we tried to look into the genome of SYNC3A and also Capricolum to find some nucleases, and especially membrane nucleases. And the only nuclease that we found that was possibly buried at the membrane is ribonuclease Y, which is related to the degradosome. So it's like nucleases related to the turnover of RNA inside the cell. And those are like essential genes for both MCAP and SYNC3A. So, but we were not able to find specifically what is the culprit nuclease for this effect that we saw in the lysate. It could be any other one. We were just looking at mainly at these nucleases membrane associated, but my guess so far is that I think those nucleases are essential for the growth of SYNC3A and MCAP, which makes things more complicated, I think.
I have time for one more question.
Adding to what Andrei just said, so we have our team has. It is logical to me that for growth in a goat, the mycoplasmas would need the surface-associated nuclease to improve the import of DNA, RNA precursors. But it may be that there is a surface-associated nuclease that has evolved such that by blast, we are not finding it, but that it may be a quasi-essential gene on the surface of the cell. So Kate Adamala and I have talked about, she's planning to take the BioBricks free gene set, and we will express all of the surface-associated proteins in mycoplasma in the free gene set and see if any of them demonstrate a ribonuclease activity, and conceivably, if it is a quasi-essential gene, then we could take this thing out. Just a thought. I think it's time we move on now to our next talk, unless anyone has any last points to make.
No, I would ask a thing, but I can just write to Chris. That's just no problem. I don't want
No,
to.
please go ahead. Go ahead.
Oh, okay. So, hi, Chris. It's been a long time.
Yeah.
So, regarding the point the referees are asking about, like, are you sure it's about nucleases? Did you get to try using some modified nucleotides that could be resistant to a nuclease activity? I know that from my experience with microRNA, we have some modified nucleotide microRNAs. They are more stable, but I don't know if, for instance, like aptamers would still work if you do that. I don't know if they can be transcribed if you think about a coding sequence, but maybe the aptamers would work.
Yeah, I'm not exactly sure about the aptamer folding using modified bases because I do know the secondary structure is
Yeah.
very important to it actually making that binding pocket for the ligand. So I know we haven't tried anything like that, no. But that might be interesting. Yeah, you might have to send me what modified bases you're actually using. Be something to look into.
Yeah, because I know I've never used them, but I read the literature because I worked with microRNAs and they use that a lot. And I mean, at least for microRNAs, they're still able to recognize their seeding sequences in the targets, and they are able to pair with the targets, even the modified nucleotides. So I'm not sure how it could interfere with secondary structure, but maybe it's worth giving a try. It's probably not that expensive. Like aptamers are small, so just give it a shot.
This is something that we are kind of trying here in the lab now. I saw one paper about it. It's like these methylated nucleosides. They are a bit more stable in like mediums contaminated with mycoplasma or something like that, right? So yeah, this is something that we are looking into now. So hopefully we'll get some good news from that.
Good.